microphone here because this podium's too short for me. Our next speaker is from Wake Forest Institute of Regenerative Medicine. He's going to be speaking on tissue engineering of liver using decellularized scaffolds. Please welcome Shay Soker.
I'd like to thank Aubrey for giving me the opportunity to present our work here. Am I switching the slides, or you do? All right. So just a little bit of where I'm coming from. Wake Forest Institute for Regenerative Medicine is in Winston-Salem, North Carolina. The mission of the institute is to improve patients' lives by developing regenerative medicine therapies and support technologies. The institute currently is about 300 people. Of these, 30 are faculty members, about the same number administrative assistant, and the rest are research scientists. The institute may know best by Dr. Atala, the director of the institute, who's wearing the director's hat or funnel. So the institute is not only doing tissue engineering, as you see here, we're also developing cellular therapies. And then in parallel, we're taking the technologies that we're developing for diagnostics platforms, other healing therapies, and then enabling or supporting technology that can apply into other areas. And as you can see here, there are sort of multiple small programs that are joined together in this path for a preclinical and then FDA-approved GMP manufacturing to get to a clinical product. So each of these units can create its own project that would go through this clinical translation, or we can take biomaterials and bioprinting, as the previous speaker talked about, and put them together and run them through the same scheme. The Winston-Salem area is now going to develop the area where the institute is, around here, as the largest urban research park in the United States, and Winston-Salem was ranked as number 18 as the best place to live in America. So back to Cambridge. I had the opportunity to visit here in the summer. It's a little warmer, it was then, and one of the activities that we were we said we were suggested to do was punting on the river, and you can probably see this sort of great guy that we had that in one hand can hold the punt pole, and you know what's the other hand holding. So going back to our business of aging, and the question that I guess we're all asking ourselves is, why are we aging? And this is some pictures from Bicentennial Man, where actually they had to use 3 different actors to actually get Haley Kate Eisenberg to grow and to age and to get to an adult and an age woman. Whereas Robin Williams as the robot stays the same throughout her life. And if you want to imagine Robin Williams as an older person, this is what it is. And these are some of the reasons why are we getting old. And I think that what we're actually discussing here in this conference is the normal aging, but we have to compound this sort of normal aging with injuries, degenerative disease, and so on and so forth. So what does the current medicine have to offer to all of these diseases? Basically, not a cure, but a treatment that would sort of delay the deterioration. And it works in many cases. We have medications for everything you want, from a headache all the way to a kidney failure. But if everything fails, medicine is resorted to organ transplantation. And organ transplantation is great. You know, this is more than 60 years with great sort of advancement of medicine and patient extending of patient life. But for organ transplantation to exist, actually the donor need to give up the organ for the recipient to have a leg. And this is basically a zero-sum game. You know, one has to lose for the other one to win. So that sort of has resulted with this sort of disparity between the number of transplanted organs and the number of people that are waiting for transplantation. And as you can see, and I didn't even bother to put the years here, but we know that this gap is growing. So there's not enough there are not enough organs to replace damaged or missing organs. So the question, and the question that in Bicentennial Man they've asked, I mean, can we actually fix organs? And they actually could fix the robot's heart. But we can replace those organs. I mean, we can have a bionic leg, or a Captain Hook can have his hook. But imagine if Captain Hook's mom wanted him to go and play piano. He wouldn't do any of the two hands piano pieces. So as good as these artificial organs are, you know, take for example the artificial heart, there still have significant deficiencies. The body sees them as foreign agents. As good as they are, they can still not duplicate exactly the native organ, and they wear off. So the idea was, can we make organs in the laboratory? And that idea is very old. Here, Charles Lindbergh, who is known for other things, teamed up with Alexis Carrel, a very talented scientist and a physician, to make the heart perfusion system here. And then they went on to actually write a book on the cultures of organs. And that was in 38. About 50 years later, Vacanti and Langer had actually created the first human ear, and the first human ear in a mouse. So all of that is great. I mean, we've advanced the field of tissue engineering, but you can all see that the mouse doesn't hear from this ear, but it hears from its own ears. So we still have a way to go, even though we had 50 years of research. And the presentation that we've seen just now actually talked about new technologies to make these things better. So we sort of tasked ourselves with engineering functional organs. And this is a primer on tissue engineering for those who are not in the field, where we always start with cells, because these are the building blocks that we use from a biopsy. We place them on the scaffold, and then we see that the tissue is formed. And then once we confirm that, we actually take it all the way into creating an organ. This is the example of the bladder, the first whole organ that was engineered. This was when we were still in Boston. And we followed sort of this presumably simple diagram where in the bladder there are two main cell types: the urothelial cells that line in the inside of the bladder, the muscle cells on the outside. Seed them sequentially, and then you can have an organ in a beaker, and then that organ can be transplanted into a patient. But what my talk today is really focusing on the following. So if tissue engineers are trying to replace organs, and by taking cells and seating them on structures or scaffolds, these scaffolds should be be able to support 3D tissue formation, and B, mimic the natural microenvironment. And with everything that was done, and that year was done using a polymer called polyglycolic acid that you can see here, and at the higher magnification, it looks pretty nice. It has channels in it, it has a 3D topography, but if you look, let's say, at a vascular structure from a living sort of organ, you can see that this structure cannot mimic the microstructure of the vasculature. More so if we're talking about the liver, obviously to expect that this very uniform polymeric structure to actually recapitulate all the information in the liver was still far away from that. So our approach was to use tissue as a scaffold because it provides the authentic structure that support the cells. It has some bioactive molecules, and I'll show you some example. And it provides a reservoir of biological signals that are difficult to replicate by a generic polymeric material. And I'll use the term extracellular matrix or ECM. And this is basically the technology. If this is the tissue, and these are the cells, these are the fibers, in many cases collagen fibers, through a process that we call decellularization using detergent, remove all the cells, and then we're left with the scaffolding material, with the shell around the cells, and then we can reseed this shell with other cells. In many cases, this would be an animal organ that will be seeded with the human, whether autologous or allogeneic cells. This technology was first published by Doris Taylor from the University of Minnesota. She decellularized a whole heart and was able to perfuse back rat neonatal cardiomyocytes, and she got a beaten heart. A few years later, a group from London was able to take the same approach into the clinic by taking a trachea, removing all the cells from the trachea, taking biopsies from the patient, a bone marrow to make chondrocytes, bronchial epithelial cells to make the inside of the trachea. And then after a few days in the bioreactor, you have a trachea was transplanted here to replace this strictured trachea, and then you can see here the lung is cleared from all the liquids, and this was published in 2008. Martin Birchall and Paolo Macchiarini. So the approach can be taken from the lab into the clinic, and what I'll tell you now is about our work on liver. So we can harvest livers from various animals, various size. We've used mostly mouse, ferret, and pigs. The liver is cannulated here, and then first washed with water, and then perfused with detergent. We use Triton, a mixture of Triton and ammonium hydroxide, and you can see that within 2 hours, all the cells are removed, and we have a transparent extracellular matrix scaffold that preserves the structure of the liver. So the first thing that we wanted to see is some of the ultrastructure, see if that's being preserved. And you can see here, this is the portal triad, the portal vein, and bile duct and hepatic artery. They look preserved, and at a very high magnification, you can even see the collagen fibrils. This staining just to show that we are left with extracellular matrix without any remnants of cells. The next thing that we wanted to test is how intact the vasculature is, because one important feature of these decellularized organs is that they can be connected to the circulation of the patient. And if they're going to leak, it's not going to have this advantage. So what we did was injected a fluorescent fluorescently labeled proteins, or in this case, this was dextran, sugar of a 220 kilodalton. And what we could see is that the fluorescent beads remained within the vascular structure. Even at the higher magnification, we can see that they are filling up the small capillaries, but they are not diffusing. And then we could actually reconstruct this structure by confocal microscopy and reconstruct a native liver capillary network. We've done the same thing in a pig liver using contrast agent, and again, the vasculature is intact. We've done some characterization of the extracellular matrix just to show that we are preserving most of the components of the extracellular matrix. Now the cells are gone, and I won't elaborate too much on that, just to say that the main extracellular matrix component, the collagens, the laminin and fibronectin, are all preserved the same as the native liver. So if the extracellular matrix is preserved, does it have a function? To test the function, we took hepatic progenitor cells. These hepatic progenitor cells can give rise to hepatic and biliary lineages, and we grew the cells on this scaffolding material under two conditions, growth conditions and differentiation. What we could see is that EPCAM, a marker of progenitor cells, is highly expressed in these cells. Actually, we select them using EPCAM, so this is not surprising. And they also express the fetal version of albumin, alpha-fetal protein. When we switch the medium to differentiation medium, you can see that the EPCAM now moved to the membrane, the same as you see it in mature hepatocytes, and now we have an increase in albumin expression and shut-off of alpha-fetal protein. So this extracellular matrix provides the information for progenitor cells to become hepatic cells, and I'll show you an example of the biliary in a few slides. So now we have to take this scaffold and stuff it with cells. This is a little complicated slide, but what I'm going to tell you is that we can do it by perfusing the cells through two of the main ways to enter a liver: the central vein and the portal, the vena cava, which would feed into the central vein, and the portal. And what you see here is if we go from the vena cava, which is retrograde, we will deposit the cells in the center of the liver hexagonal lobules. Whereas if we go into the portal vein, it will be deposited on the outside. And this is exactly what you see. When we take labeled cells, fluorescently labeled cells, we put them through the vena cava, they line up the center, we put them through the portal vein, they deposit in the periphery. And actually this is an electron microscopy showing 3 endothelial cells lining a vascular structure. The next thing we did was to perfuse blood into a endothelial cell seeded scaffold. So what you can see here are the red blood cells. But what's important to see is whether this seeded scaffold can be anti-thrombogenic. And the answer is here. You can see this is staining for integrin alpha 2b, which stains platelets. And what you can see here, if we have a seeded scaffold here, no platelets, but here are the cells that we've seeded. Whereas if we have an unseeded scaffold, no cells. These are the cells. These are the nuclei of the cells that we've seeded. Many platelets and no cells. So the conclusion here is that a seeded scaffold can be anti-thrombogenic. And in parallel, we check the secretion of prostaglandin by induction with bradykinin, and we see that the cells are secreting anti-coagulation factors. The next thing was to seed the scaffold with both endothelial cells and hepatic cells, and this is, to our surprise, a pleasant surprise. What we saw is that the cells can actually find their own niche. This is the staining for endothelial cells, and this is the staining for hepatocytes. And these were co-seeded with endothelial cells and hepatocytes. The endothelial cells finding large and small vascular structures, whereas the hepatocytes, the big sort of pinky cells here, are in the parenchyma. Moreover, after 7 days in a bioreactor, we start seeing staining specific for bile duct. So the information encoded by this scaffold can allow the progenitor cells to differentiate into bile duct, hepatocytes. This is STIP 450, and we usually add endothelial cells in addition to these hepatic progenitor cells, and the cells can localize to where they need to be. And looking at urea secretion and albumin secretion, it is higher in the scaffold than if we compare it to cells in culture. Last slide is can it work in transplantation? We took a scaffold. This is an unseeded scaffold. You see, it's white, it's not yellow. And we connected it through the portal vein and then into the vena cava. This is the clamp, and see what happens when you remove the clamp. Blood is flowing into the scaffold, no leakage. Indeed, this scaffold was clotted because it had no endothelial cells, but right now we're on experiments with seeded scaffolds, implanting them in rats. So what's next? We're taking this technology and working on kidneys, pancreas, and intestine in order to engineer these organs as well. Thank you, and I'll be happy to answer any questions.
We have 2 minutes for questions.
Michael.
So you start off your talk talking about how it's a zero-sum game to do conventional transplant medicine. There's a series of challenges even to turning the decellularized matrix paradigm into something other than a zero-sum game, because you still have to get the original matrix from somewhere. And obviously you don't want to take it from someone who's alive right now as you do with conventional transplant medicine, so you have to take it to someone who's deceased. And we already have a very low rate of people being willing to give their organs even for regular transplant medicine. And also you tend to get people who are progressively older, and as we discussed last time we were talking about this, the ECM itself has all these structural and functional properties that degrade over time with aging. So in a real transplant situation, as opposed to in a mouse situation where you can choose a young and fully functional organ to decellularize, you're going to have, dubious full supportive viability. Now, I'm not saying any of these are deal breakers, but I'm just, a lot of them are outside of your actual narrow scientific work on the subject, but how do you envision overcoming that series of challenges to actually make it so we're not back with a different zero-sum game?
Right. It's a good question, and I can give you several directions that we're going after. Obviously, the first one would be to harvest it from animals that have a similar, that their organs have a similar anatomy to humans. So right now we're using the pig as our model for harvesting organs. But even if you if you see how many organs are being rejected for transplantation, imagine that we can take those organs, and this is probably, I haven't looked at the literature, but I think that of 5 organs that are being donated, anywhere between 2 or 3 are being rejected. So if we can increase this number of organs that are available for transplantation even by 2-fold, we actually are making maybe a slight dent in that increasing gap between the available organs to and organs that are offered. So whether we go for animals or just using all the organs that are being transplanted, these are sort of two approaches that we're looking at. And we may not be able to actually produce organs as a supermarket, but at least we'll be able to provide more than what's currently available.
What is the nature of the communication between the scaffold and the cells, and will the scaffold itself be immunogenic to the acceptor?
So we have not seen any rejection except for slight inflammation that goes away when we implanted empty scaffold in animals. On the contrary, what we're seeing is that we have some mobilization of mesenchymal stem cells that are actually helping in sort of the healing process. So that's how the host is treating or interacting with the scaffold. In terms of the cells, we've in different scaffolds that we've made from different organs, we've always seen that the scaffold provide a much better substrate for cell function, not so much for cell differentiation. Cells do not proliferate on these scaffolds as well as they do in tissue culture plastic. But the function has always been much better than in plastic.
But you don't know anything about the signals between the scaffold and the cells?
We believe that these are the natural signals, let's say integrin binding, that the natural extracellular matrix provides to cells.
Do you somehow help the progenitors to differentiate in the types of cells that you want? Do you add some growth factors or are the small molecules?
Sure, yes. By itself, we see some differentiation, but we can't get to this functionality that I've shown you.
You used endothelial cells and hepatocytes, but are more important cells, the liver, like Kupffer cells and fat storage cells. If you want to have a complete intact liver, you should use those cells as well.
Well, yeah, and that's, you're absolutely right. Our approach is to use hepatic progenitor cells that can give rise to many, may not be all of the liver cells. What I haven't shown you is a set of experiments that we're doing now where we're adding the stellate cells that are also very important for the endothelial and the hepatic interaction. So true, I mean, our goal is to recapitulate at least the minimal metabolic activity of the liver and if possible, the biliary activity.
So you said you were working on intestine, and I was wondering that made me think of the crypt stem cells that occupy the villi and how they normally proliferate and maintain homeostasis of the intestine. Have you looked at the reestablishment of stem cell populations in the stem cell niche in some of these organs, whether or not they're reestablished, and that might the viability of the
No,
organ?
we have actually we've just written a grant on looking at the stem cell reconstitution of a stem cell niche using in a liver using this model.
And second question, have you considered using organs that have been stored for months in formaldehyde? Is that possible?
Well, the concern will be that they will be over cross-linked and then they will not be remodeled the way that these are. Because what we think is that upon cell seeding, cells continue to secrete enzyme and extracellular matrix and remodel this matrix to actually fit. I mean, this is, in our case, extracellular matrix derived from animals. And there are differences between these animals and humans. So the cells will have to adjust their environment.
We have time for one more question.
This comes back to the first question that was asked. Given that most hepatic pathology in humans doesn't affect all 8 liver segments, do you see potential to use scaffolding from the healthy liver segment and using it in the same liver that is part of the liver that is diseased?
This is actually what we'll try to do in our pig models. The problem that I may have sort of not pressed on so much is to keep the vascular network intact. Because if we cannot, if we are implanting it and not transplanting it, if we are not anastomosing the vascular network of the organ into the host, then we haven't done as much as we wanted to do. So as long as we can provide, and right now we're actually doing that, we're working on lobes and not the entire liver to be able to do exactly what you just said.
Thank you, Shay.